Very warm welcome to our learners, and today the topic of my talk is epigenetics and cell differentiation. We know that epigenetics, the difference between genetics and epigenetics is that when we talk of differentiations or variations due to genetics, it is about change in the DNA sequence. But when we talk of epigenetics, and it is causing some phenological differences in the morphology of an organism, it is without any change in the sequence of the DNA. So what makes it different in terms of expression, we will discuss it today. In the beginning, we will discuss some of the definitions so that it becomes easy for us to understand the rest of the talk. Cells of a multicellular organisms are genetically homogeneous, which means all the cells that we have in our body, they have the same genetic makeup. But structurally and functionally, it is heterogeneous. What does it mean? That when we see that the same genetic makeup is making an epidermal layer at some point of time, making skin, and the same time at other places it is making bone cells. In some other places in brain, for example, it is making neurons. So all these tissues are structurally and functionally different, and all these differences are stemming out of the same genetic makeup that we inherit, every cell of the organism inherit, and it is because of the differential expression of genes. We will learn gradually how the gene expressions differ from one tissue to the other. Many of these differences in gene expression arise during development and are subsequently retained through mitosis. So when these epigenetic changes occur in the genome, then they are stable changes. Whatever changes they bring in terms of gene expression, they remain as such and are transmitted from one generation of the cell to the next through the process of cell division called mitosis, or this is also called equational division. Stable alterations of this kind are said to be epigenetic because they are heritable in the short term but do not involve mutations of the DNA itself. We have already said that the epigenetic changes do not involve changes in the sequence of the DNA. Epigenetics refers to the study of changes in the regulation of gene activity and expression that are not dependent on DNA sequence. You see, we have to understand first the basic premise of genetic diversity or the genetic makeup of an organism is that whatever we have inherited, it remains constant throughout the life cycle of an organism. Each nucleotide remains at its own place. There is no change in the DNA sequence. While epigenetics often refers to the study of single or a set of genes, but epigenomics deals with the global analysis of the epigenetic changes across the entire genome. So we have been saying the same thing again and again, that the genetic mechanism involves the primary DNA sequence and changes of mutations in this sequence. This is called genetics or genetic mechanism of the changes, whereas epigenetics mechanisms do not involve a change in the DNA sequence. And epigenetic regulations involves the modification of DNA and proteins associated with DNA, which results in the changes to the conformation of DNA and accessibility factor. Here I would like to show you a cartoon of basic internal organization of a eukaryotic cell. You see, here we have a cross section of a cell. This is nucleus, these are cell organelles, and from the chromosome, this is one of the chromosome, and chromosome is highly compact structure, but when it has to express itself in terms of gene expression, then it relaxes itself, it opens up and allows various proteins and other transcription factors to interact with the DNA, and which eventually lead to the gene expression. So this is a chromosome, and one thread of it is composed of smaller units which are called nucleosomes. These are nucleosomes, and what is one nucleosome is that about 146 nucleotides long DNA wraps around the 8 basic proteins which are called histones. These are histones, and these DNA are wrapped around these histones, and when we further expand these threads, then we come to this helical structure of DNA, where these are the sugar phosphate backbones, and in between these green, red and blue and yellow are shown as if they are nitrogenous bases. What I am trying to say is that this DNA sequence does not change in the epigenetic modifications, but it is the major groove of the DNA. Here, for example, this. Here there is a nitrogenous base called cytosine. At the 5th carbon position of the cytosine, a methyl group is attached. And this is one of the very simplest form of DNA modifications which govern gene expression in the DNA. So, the DNA undergoes modification, histone undergoes modifications, that is not methylation but that is acetylation. We will discuss briefly about that, but we will be mainly concentrating on the methylation of the DNA. So, DNA methylation is a modification to DNA that occurs through the addition of a methyl group to DNA strand itself, often to the 5 carbon position of a cytosine ring. Since it is upon the DNA, there is a DNA molecule, on this a methyl group is sitting, therefore it is called epigenetic modification, and can be either removed through demethylation and without any disruption to the original DNA sequence. Here is a cytosine ring, and these methylations are usually, this is very important to understand, that every cytosine of the DNA is not methylated. It is the cytosine which is present in contiguity to a guanine. In our jargon we say that it is the CPG island, which means cytosine, phosphate and guanine island. Whenever a DNA sequence has got these CPG and CPG in opposite orientation, the cytosine here will be methylated. This is the guiding principle of the DNA methylation. This shows that these are our histones, DNA are wrapped around, and these brown structures are the methylation. That means the cytosine is methylated here, and the yellow structures here in the second panel here, you will see these are acetylations. Now here you will see, it is very evident from the figure, that the methylated DNA is a compact DNA, whereas acetylated DNA here is a relaxed DNA. Hence, we can assume that methylation leads to the shutdown of the gene expression, and the acetylation leads to the translation or transcription and followed by translation. So, unmethylated DNA here, but which is acetylated, leads to opening up of the histone from each other and allows accessibility to the protein and various transcription factors to interact with the DNA here, and this interaction makes it possible for DNA to transcribe. And one of the leading interacting protein is RNA polymerase. So this is the reaction here, that is S-adenosyl methionine is acted upon by DNA methyltransferase. This is one of the 4 enzymes that are operating for methylation, leads to SAM, and this methyl group is attached to the 5th position, which is then it is called 5-methyl cytosine. Let us understand why we are discussing epigenetics is, as I told you in the beginning, that despite the fact that we have the same genetic makeup, and yet every cell has differentiated into a different tissue. It is performing a different function. And how is it happening? There are, if you have studied the nucleus, then you will understand that the entire genome is divided into 2 dense and less dense components, the entire genetic material. The denser component is called heterochromatin, and the lighter stained lightly stained component is called euchromatin. And heterochromatin part is mostly the one with compact, here you see, with compact DNA, and euchromatin, which is lightly stained structure, is a relaxed DNA. So we can imagine a picture that it is the lightly stained genome or the genetic component of the nucleus which is actively transcribing, which is active, which is accessible to various protein factors and is actively transcribing. This dense component, which is highly methylated, is not actually transcribing. Transcription means the proteins are being formed and we are constantly using those proteins in various metabolic pathways. So these epigenetic mechanisms operate at three different levels. One is the DNA methylation we have discussed, and the other one is histone modification, we will briefly discuss it, and the third one is RNA interference. Now, without going into the details of the gene expression, these RNA interfering molecules, let me tell you very briefly, are the complementary copies of the mRNA, which is carrying the information from DNA to protein. So when these mRNAs which are carrying informations are formed, the interfering RNA molecules, which are relatively smaller, which are complementary to those mRNA, they join together and render them inactive. So RNA, since it is interfering in the functioning of the genetic machinery, therefore they are called RNA interference. RNA interference, they interact with histone modification and they interact with DNA methylation, but the major interaction takes place between histone modification and DNA methylation. And all these three major activities of the cell have an effect on gene expression. Here is a cartoon which shows that the upper panel here, in which these cytosines are not methylated, although they are CpG island, and yet they are not methylated, therefore they are actively transcribing. Metabolically they are active, but the moment they are methylated, shown with the red circles, they become silenced. Now these are, I was telling you, DNA methylation, histone modification, which includes acetylation and methylation, non-coding RNAs, microRNAs, which are about 22 nucleotide long mRNAs, and all these three regulate gene expression. These are the enzymes which are responsible for the methylation. DNMT1 and DNA methyltransferase 3A is responsible for the maintenance of established pattern of methylation pattern, and 3B, they establish de novo methylation pattern. That means they can find any cytosine which is present in the CpG island, it will methylate them. There are two additional enzymes which are DNMT2 and DNMT3L. They have the similar function. Now, these epigenetic changes are often associated with the syndromes that are found in the human population. Let me tell you that not all genes are active at all times. We have been harping on this theme that euchromatic and heterochromatic regions, they change from time to time. So that means all the genes are not active all the time. They are regulated, they are switched on and switched off. And DNA methylation is one of the several epigenetic mechanisms that cell employ to control gene expression. And therefore it is an important component in the cellular processes, including embryonic development, genomic imprinting, X chromosome inactivation, which is the formation of Barr bodies in the female cells, preservation of chromosome stability, heterochromatin maintenance, tissue specific expression controls. It is not surprising that the researchers have also linked errors in methylation to a variety of devastating consequences, including several human diseases. So any variation in the wrong methylation of certain DNA can also lead to disease conditions. That is why it is important to study the methylation pattern and the correct methylation pattern. This methylation pattern, since it is methylating the CpG island, and CpG islands are found in the repetitive portions of the DNA, and also they have been located in the 5' side of the promoter region. You know, the gene in the eukaryotes is a split gene. That is, it has got introns and it's got exons, and all these intron exons are driven by a common promoter at the 5' end. And these CpG islands are mostly located in the promoter region. And promoter region is the one which interacts with the RNA polymerase for the transcription of the downstream and structural genes in an operon. So cancer has also been implicated and has been shown to have links with the pattern of the methylation pattern of the DNA. So we'll take one example, the loss of tumor suppressant gene. We have number of tumor suppressor genes have been studied, and one of the most studied gene is P53. So TSG is the tumor suppressor gene. It is a kind of a master gene which is masking the effect of the proto-oncogenes, which are actually cancerous genes, but these genes remain suppressed because of the presence of the wild type of P53 gene. So this is also called a tumor suppressor gene. That means it suppresses the tumor causing genes. In short, they are called TSGs. Many such genes which have a masking effect have been identified. P53, P16 and KI1 are one of the common, and P10 are some of the common examples which are being studied. There is a cartoon again here suggesting this is a hypothetical piece of DNA which has got several genes in it. I would like to draw your attention to this portion which is shown in red line, which is the part of the promoter gene. In an operon there is a promoter gene, there is an operator gene, and then followed by structural genes. Promoter gene is the one which determines the direction of the transcription by showing asymmetry in the conserved region, that is -10 and -35 regions. That is why RNA polymerase comes and sits in a particular orientation, and then it can move only in one direction, and that is the direction in which it passes through the operator and into the domain of structural genes. So here I would like to show you that this CpG island is a part of the promoter gene, which is immediately followed by tumor suppressor genes. Here, TSG. This bent arrow shows the direction of the transcription. Now these are empty cells, therefore they are hypomethylated. That means they are not methylated. And if you recall from my previous slide, that methylated portion prevents gene expression, whereas hypomethylated or unmethylated portions they promote gene expression. So this is not methylated, therefore it promotes the expression of TSG. It is hypermethylated. So transcriptional repression, loss of TSG expression, and the moment they are methylated, if they are not methylated, this is working all right. But the moment they are methylated, they become inaccessible to RNA polymerases, leading to transcriptional repression, loss of TSG function, loss of function, leading to cancer. This has been shown in one of the experiments that links epigenetic modifications to cancer. That is, epigenetic means DNA methylation to cancer. One more thing, here you would like to understand that these repetitive sequences are located near centromere. So they are usually called pericentromeric heterochromatin. Some of the human diseases which have been associated with DNA methylation are imprinting disorders, repeat instability diseases, and defects of the methylation machinery, leading to complete shutdown of the metabolic pathway, leading to various abnormalities. Now, we will come to the second part of our talk, which will deal with the cell differentiation.